The University Hospital of Clermont-Ferrand is divided of three main hospitals: CHU Gabriel-Monpied in the south, Louise Michel in the north, CHU Estin in the limit of the city centre. Estin Hospital was built in 2010 as part of the urban regeneration project of Clermont-Ferrand. It replaced an old hospital based in downtown of Clermont-Ferrand and located on the site of a former Michelin factory. And now the University Hospital of Clermont-Ferrand has a total capacity of approximately 2,000 beds.
Hello everyone, I'm Aurelien Lebreton. I'm the head of the Hematology Laboratory Department in CHU Clermont-Ferrand, France. I'm also the head of the Hemophilia Comprehensive Care Center of our university hospital. About our laboratory department, we have two sites, Gabriel-Monpied and Estin. We have some analyses that we perform 24 hours a day and 7 days per week, and we have also some specialized analyses that are only performed on working hour in our hospital. About some coagulation assay performed in emergency, we are able to respond to our partners 24 hours per day for some emergency analyses, such as analysis for the diagnosis of bleeding coagulation, bleeding disorders, or also some anticoagulated patients, and perioperative management of hemostasis. Yes, we have some topic where we are involved in research. Our main part is the study of the impact of cirrhosis on coagulation, and we have also in a recent decade developed a unique platform dedicated to the evaluation of new drugs or new system performing some coagulation assays. And this platform allows us to develop several collaborations with different partners. Some of partners are involved in the medical and pharmaceutical industry, and also we have some collaboration with IVD manufacturers such as HORIBA. Yes, of course, D-dimer is always a hot topic. D-dimer is a routine clinical evaluation of exclusion of venous thromboembolism, for example, deep vein thrombosis and pulmonary embolism, but also has some other indications, such as the diagnosis of disseminated intravascular coagulation. And in recent years, we have some new interests such as COVID-19 pandemic or aortic dissection, or also some other indication in the field of cardiovascular diseases. We evaluated in the past some reagents for D-dimer, and more recently we had a partnership with HORIBA to evaluate the performance of new Yumizen G DDi. And the main advantages of this reagent is to be liquid, to be ready to use, and have a stability on board of 2 weeks. We evaluated analytical performance of Yumizen G DDi performed on Yumizen G 800 coagulometer, and we found that this test has a good repeatability and reproducibility according to RICO standard and accreditation requirements. Close to the threshold, clinical threshold of exclusion of 500 nanograms per milliliter FEU, we found that Yumizen G DDi have a satisfactory agreement. In this study, we did not find a significant interference with bilirubin and hemoglobin. We also evaluated the impact of rheumatoid factor and human anti-mouse antibodies, and we did not find any significant interference with Yumizen G DDi. All of this work about clinical performance was presented at the International Congress of ISTH in London and was published in International Review in Research Practice on Thrombosis and Hemostasis. I think that it's really important to use in our lab some D-dimer reagents that are analytically performant and well evaluated, and also it's really important to have some clinically validated D-dimer reagent.
HORIBA.